Hello everyone and welcome to our module on virus structure. Let's start by reviewing some of the basic features of viruses. Viruses contain nucleic acids, either DNA or RNA, and these nucleic acids are surrounded by proteins that make up a structure called the capsid. Now in some viruses the capsid is also surrounded by another structure called the envelope. And then importantly, viruses have no metabolic activity. Their only goal is to transmit their nuclear material into host cells so that that nuclear material can be reproduced into more viruses. So here's a picture of the basic structure of a virus. There are nucleic acids in the center, that's called the genome. Those nucleic acids are surrounded by a capsid, and then in this case the capsid is surrounded by an envelope. And then the envelope often contains glycoproteins, which are important for attachment to host cells and infection of host cells. Most capsids have one of two common shapes. The first common type is a helix. This is a helical capsid drawing on the right side of the screen. The other common shape is an icosahedron. This is a drawing of an icosahedral capsid on the left side of the screen. An icosahedral capsid looks kind of like a soccer ball. And both of these structures are made up of multiple identical copies of the same protein, and that's the way viruses like to make their capsids. They synthesize one protein and put multiple copies together to form either a helix or an icosahedron. Next, let's talk about envelopes. So viruses are called either naked or enveloped, depending on whether or not they have an envelope surrounding their capsid. And the envelopes are lipid membranes that are acquired from the host cell during assembly of the virus. Usually the virus acquires this from the cell plasma membranes, but there are some exceptions. Some viruses acquire their envelope from the nuclear membrane or the endoplasmic reticulum. There are often viral glycoproteins embedded in the membrane, and these can be used so that the virus can bind to host cells. There are also antigens for the immune system. And one easy way to remember which viruses are enveloped and which are not is to know that all naked viruses have an icosahedral capsid. The icosahedron shape is very stable, so if a virus is going to lack an envelope, it is going to want this icosahedron shape to keep it stable. Viruses that have an envelope may be icosahedral, but sometimes they're helical as well. A rule of thumb that can be very helpful to remember is that in general, envelope viruses cannot survive in the environment, so they usually are not transmitted fecal-orally or through contaminated drinking water or anything that would require them to survive for long periods of time outside the body. So for example, hepatitis B is an envelope virus and it's transmitted sexually. The herpes viruses are all transmitted through direct contact, and HIV, is transmitted sexually. And this makes sense because they're all envelope viruses and they're not going to survive very well out in the environment. On the other hand, naked viruses have a durable protein coat, so they often can survive out in the environment. So adenovirus, for example, survives on surfaces. Rotavirus can be transmitted fecal-orally. Hepatitis A and E are found in contaminated drinking water, and rhinovirus also can survive in the environment. So remember this rule of thumb, and it can be very helpful for remembering the structure of viruses as long as you know the way by which they are transmitted. I told you before that viruses carry nucleic acid genomes that are made up of either RNA or DNA. So let's talk about that in a little more detail. And let's begin by discussing the RNA virus genomes. So most RNA viruses carry a genome that's made up of a single strand of linear RNA. There are just a couple of exceptions to this general rule. The reoviruses are very unusual in that they carry double-stranded RNA. And then the retroviruses are sort of an exception. Most textbooks will tell you they have single-stranded RNA for their genome, and that's true, but they have two single strands. They're diploid. And then I said that most RNA viruses are linear. The exception to that goes by the mnemonic BAD. The Bunya viruses, the arenaviruses, and the hepatitis D virus or the Delta virus, those all have circular genomes. And then most RNA viruses replicate in the cytoplasm. That's where mRNA gets transcribed by eukaryotic cells into proteins. So it makes sense that most RNA viruses would want to replicate there in the cytoplasm. The two exceptions to this are the influenza virus and the retroviruses. These replicate in the nucleus. You need to be familiar with the concept of RNA virus polarity. So we designate RNA viruses as either positive sense or negative sense, and we call this the polarity of the RNA virus. RNA viruses that are positive sense or that are positive stranded carry RNA that is structurally similar to mRNA. This means that once the viral RNA gets into the cytoplasm of the host cell, it can be used immediately for protein synthesis by the host cell machinery. The host cell will interpret that piece of viral RNA as a piece of its own mRNA, and it will begin synthesizing proteins using the mRNA as a template. Negative-stranded or negative-sense RNA must first be converted to positive RNA. Only then can it be used as a template for protein. This means the virus must carry an enzyme to convert the negative-sense RNA to positive-sense RNA, which can then take over and use the host cell machinery to synthesize proteins. So let's drill down a little further on this concept of RNA polarity, and in order to do that we have to review a little bit of biochemistry. So nucleic acids are made up of repeating ribose sugar units, and this is a ribose sugar in the top right corner here. And when the ribose sugar has this little OH right here removed, it becomes deoxyribose. So DNA stands for deoxyribonucleic acids, and the ribo is for the ribose sugar, and RNA stands for ribonucleic acids, and that ribo is also for the ribose sugar. Now we number the carbons on the ribose sugar starting with the one right next to the oxygen. Let's look at this picture in the middle here. So we've got carbon number 1, and then number 2, and then number 3, and then number 4, and then number 5 is up here. So attached to the one carbon is always some base, and in this case we've shown a guanine base. And as you know, nucleic acids are made up of different bases. And the RNA bases are uracil, guanine, adenine, and cytosine, and they often just go by the first letter of their name, U, G, A, and C. So the way that the nucleic acids are built is that the 5' carbon right here is always attached to a phosphate group, which then attaches to the oxygen hanging off the 3' carbon on the next ribose molecule. And this is how nucleic acids are built, from monomers of these ribose molecules attached to bases and then phosphorylated to another ribose. But the most important point here is to understand how we name the different ends of the nucleic acid. So one end of the polymer will have a phosphate group hanging off it. It will not be attached to another ribose molecule. And that end with the phosphate is called the 5' end. Another end of the nucleic acid will have an OH group hanging off of the 3' carbon of one ribose molecule, and we call that the 3' end. So this direction from the 5' to the 3' end is very important for determining the polarity of RNA inside of RNA viruses. So a positive sense RNA virus will look something like what I've shown at the top of the screen here. There will be a 5' end and a 3' end, and between them will be a series of bases that make sense. These bases can be interpreted by ribosomes and synthesized directly into proteins. What a negative sense RNA virus will look like is something like what I've drawn here. It will have a 3' end and a 5' end, and the nucleic acids will not make sense. They will not be translatable into proteins in their state in which they're found inside the virus. However, the enzyme RNA polymerase can come in and make a mirror image copy of this negative sense RNA. It will flip the 3' end to become the 5' end, and it will change the 5' end to become the 3' end, and then it will flip each base. So as you know, G's and C's go together and A's and U's go together. So every G will become a C and every A will become a U. And once it flips all the bases, it will now have created a strand that looks just like that positive sense strand I drew up above, and that can now be directly synthesized into proteins. So let's talk about the enzyme RNA polymerase. Recall that human cells make RNA from a DNA template strand. This is called transcription. And the enzyme that does this is called DNA-dependent RNA polymerase. So this enzyme is going to synthesize RNA polymers, that's the RNA polymerase part of the name, by depending on DNA as the template. Viruses, on the other hand, need to make RNA directly from RNA, and there's no readily available human enzyme to do this, so the virus has to bring or synthesize its own enzyme, and this enzyme is going to be called RNA-dependent RNA polymerase. So it's going to synthesize RNA polymers, but it's going to depend on an RNA strand as the template as opposed to the human cells which depend on a DNA strand as the template. So now let's talk about how RNA polymerase can be used by positive and negative sense RNA viruses. First, let's consider the case of a positive sense RNA virus. It will bring in a piece of RNA that looks just like messenger RNA, so the host cell ribosomes in the cytoplasm will immediately begin using it as a template for the synthesis of proteins. One of the proteins that will be created will be an RNA polymerase, which will then make a negative sense copy of the positive sense RNA that came into the host cell with the virus. RNA polymerase will also then use the negative sense RNA strand as a template to create multiple positive RNA strands that are the viral genome. Now you've got lots of viral proteins present and lots of viral genomes present, and these can all be packaged together to make more virions that can then be released, and this is how the virus infects cells and multiplies. The case of a negative sense RNA virus is a little bit different. So a piece of RNA with negative polarity infects the cell, and because it has negative polarity, it doesn't look like mRNA, and it can't immediately be used by the host cell ribosomes in the cytoplasm. This means the virus must bring with it its own enzyme RNA polymerase in order to begin reproducing itself. That enzyme RNA polymerase will make a positive copy of the RNA. So now we've got a positive strand of RNA that looks like messenger RNA. Now those ribosomes can convert this into proteins, In addition, that positive sense RNA strand can be used by RNA polymerase to make multiple copies of the negative RNA strand, which are the viral genome. Now you've got proteins present, you've got viral genomes present, and you can package them together into new virions. There are two RNA viruses which are retroviruses. The most famous of these is HIV, but there's another one called human T lymphotropic virus. And both of these viruses have a genome that's composed of two strands of positive sense RNA. These viruses carry an enzyme called reverse transcriptase, which uses the RNA as a template to make a double strand of DNA, just like we typically find in human cells. These then go through the normal process of transcription inside the host cell to be converted into mRNA, which then uses ribosomes in the cytoplasm to create copies of the protein. So these have a very unique mechanism by which they reproduce. Now let's talk about viruses that have DNA for their genome. These viruses have either a circular strand or a linear strand of DNA inside their capsid, and most viral DNA genomes are double-stranded. The one exception is parvovirus, which has only a single strand of DNA. You should be familiar with the concept of viral genome infectivity, and this refers to whether or not the nuclear material of the virus by itself can cause infection. So positive sense RNA genomes are infectious by themselves. Remember, those genomes look just like messenger RNA. So the genetic material begins producing new proteins, and using those proteins it makes new nucleic acids as soon as it gets into the cell. There are no extra enzymes required, just the nuclear material itself inside a cell can lead to production of more viruses. The same thing is true for double-stranded DNA genomes. These are infectious by themselves, because if they get inside a host cell, there is machinery already present in that host cell to start using them as templates to make proteins and new virus particles. Negative sense RNA molecules, on the other hand, are not infectious by themselves because they require RNA-dependent RNA polymerase in order to reproduce. If you get one of these inside a cell, but you don't also put the viral enzyme with it inside the cell, then no infection will occur and no new viruses will be created. Many RNA viruses have what's called a segmented genome. This means that the nuclear material exists as multiple molecules of RNAs or segments inside the virus. This allows the virus to undergo a process called reassortment. So imagine that two viruses co-infect the same cell. They can have mixing of their segments into a new virus. And the most important example of this is influenza virus. Influenza virus every once in a while reassorts itself, and sometimes this causes pandemics around the world. There are 4 families of viruses that carry segmented genomes, and you can remember them by the mnemonic BOR, which stands for bunyaviruses, orthomyxoviruses, which includes influenza, arenaviruses, and reoviruses. Now, most of the viruses in these families are very rare. For example, California encephalitis is very rare, Lassa fever is very rare. The 2 viruses in all these families that are relatively common are influenza virus and rotavirus, which causes diarrhea in children. So if you want to remember 2 relatively common, higher yield viruses that are segmented, remember influenza and rotavirus. On this slide, I've listed the names of all the DNA viruses, so let's go through this. So we've got naked DNA viruses and we've got envelope DNA viruses. The naked DNA viruses include parvovirus, adenovirus, papillomaviruses, and polyomaviruses. There's only one parvovirus you need to know, that's B19. There's only one papillomavirus you need to know, that's human papillomavirus, and there are 2 polyomaviruses, JC and BK. Down at the bottom, I've written the envelope viruses. These include hepadnavirus, which is hepatitis B virus, the herpes viruses, and there are quite a few of these. We're going to have a separate module on all the herpes viruses. And then finally, the pox viruses like smallpox and cowpox and molluscum contagiosum. Now, I write them in the order I've written them on this slide on purpose because there are some groupings that will help you remember things about these viruses. These 3 in the middle right here are the 3 that have a circular DNA structure. So papillomavirus, polyomavirus, and hepadna all have circular DNA. Parvovirus is the smallest of these viruses, and therefore it is the only one that has a single strand of DNA. It's the only one that's not double-stranded. Pox virus is the largest of the DNA viruses, and therefore it's the only one that replicates in the cytoplasm. I think of it as being too big to fit in the nucleus, which is where all the rest of these reproduce. I've also written some rules of thumb down here on the bottom. All the DNA viruses have an icosahedral capsid except for pox, and all of them replicate in the nucleus except for pox, which is what I said before. I've written all the names of the RNA viruses on this slide. This is a much bigger group than the DNA viruses, and these ones are much harder to keep straight because there are lots of exceptions to all the rules of thumb about their structure and how they replicate. At the top, I've listed all the positive sense RNA viruses, and at the bottom, the negative sense RNA viruses. All the positive sense RNA viruses have an icosahedral capsid except for coronavirus. That's why it has this little asterisk here. I've also written that here down at the bottom of the screen. Then there are naked icosahedral capsid positive sense RNA viruses and enveloped icosahedral capsid positive sense RNA viruses. And finally, all of the negative sense RNA viruses have a helical capsid, and they're all enveloped. The names of them I've written down the side here, and I won't go through them all in this module, but in the module on RNA viruses, I'll go through and name all these families in each of the viruses and talk about the infections that they cause. Some general rules of thumb are down here at the bottom. All of these RNA viruses are single-stranded except for the Rio viruses, which are right here. They all replicate in the cytoplasm except for influenza and retroviruses. So influenza is an orthomyxovirus, and the retroviruses are both right here. All RNA viruses have a linear genome except for the BAD viruses, and BAD stands for Bunya virus, arenavirus, Delta virus. And then finally, they're all non-segmented except for the Bohr viruses. Those are the ones that have segmented viruses. So that's Bunya, orthomyxo, and arena, and also the Rio viruses up here. I'll finish this module by discussing viral vaccines. So there are a couple different types of vaccines we use to vaccinate people against infections caused by viruses. The first type are the so-called live attenuated vaccines. These vaccines contain an inactive strain of the virus. Rarely they can actually produce clinical disease, and so you don't want to give a live attenuated virus vaccine to someone who's immunocompromised and doesn't have a strong immune system. The good thing about the live attenuated vaccines is that they provide long-lasting protection. They often require no boosters. Sometimes they do, but often they don't. And we use live attenuated vaccines for things like smallpox and yellow fever and chickenpox, which is caused by the varicella-zoster virus. There's also a vaccine for polio called Sabin's poliovirus vaccine, which is a live attenuated vaccine. The measles, mumps, and rubella vaccine, or the MMR vaccine, contains live attenuated viruses. And then the intranasal influenza vaccine, which is a nasal spray you use on children, also contains a live attenuated version of the virus. The next type of vaccines are called killed virus vaccines. So these are killed viruses, obviously, and there's no risk of infection because the virus is dead. However, these generate less immune response, so you often have to give boosters more often than with live attenuated vaccines. And we use killed virus vaccines for things like rabies. The injected influenza that you get every year if you get a flu shot is a killed virus vaccine. The Salk polio vaccine, which is another type of polio vaccination, is a killed virus, and so is the hepatitis A vaccine. And then the final type of vaccine is called a recombinant vaccine, and basically what you do here is use a culture to produce multiple copies of a viral protein that you use for vaccination. So you take a gene for a viral protein and you insert it either into a virus or some cells in culture. When the carrier virus or the cells grow, the vaccine protein gets copied, and then you can inject multiple copies of this viral protein into a person, and the immune system will recognize the expressed protein and create an immune response. And this is the way we vaccinate people against hepatitis B. We create recombinant proteins of the hepatitis B surface antigen and use that to vaccinate people. Same thing for the human papillomavirus. We use recombinant proteins of the HPV type 6, 11, 16, and 18, and then you can inject these and use them for vaccination. And that concludes our module on viral structure.